Welcome everybody to more one live of our GeneTime conference. That is a symposium of the genetic postgraduate program at the University Federal University of Minas Gerais. Today we have Suzanne Renner. She was professor at Munich University for a long time, and she's a leader on plant research. She's mother, so it's very important to say that because she can do very good science, even being a mother, a woman in this hard academic world. So, and she's will talk to us about the domestication of plants today, and I hope you enjoyed the talk. Please, Suzanne, you can start.
Thank you, Fernanda, for inviting me. I'm very sorry about this 15-minute delay. I'm sure, in fact, that my talk will be over in 30 minutes, and I look forward and I hope forward to questions, and I hope that people will stay for the end of the talk. The title I sent to Fernanda was very long: Genome Sequencing of Up to 6,000-Year-Old Citrullus Seeds from Libya and Sudan Sheds New Light on the Domestication of Watermelon. It's the title of our paper that just came out about 10 days ago. But a broader title, a better title, should have been What Ancient DNA Can Contribute to Inferring the History of Our Plant Foods. So the watermelon today, the largest producer is China. They have the most varieties, they have the most research on the watermelon, and they produce most of the watermelons eaten in large parts of the world today. But of course, China is not the place where the watermelon originated and where it was first domesticated. Instead, the wild watermelons, which is a very small genus, are all from Africa. They are all restricted to Africa. And among the oldest collections ever made are these. You see the Herbarium Specimen collected by one of the students that Linnaeus sent out all over the world to collect plants and send them back to Sweden, to Uppsala, where Linnaeus then named them. And this particular student passed through South Africa, where he spent several months, and then went on to Japan, where he spent 4 years, and then he went back to Uppsala and in fact became the successor of his professor, that is the successor of Linnaeus. And in South Africa, he did this excursion shown here and collected all sorts of plants. And in his diary, he wrote, the colonists around this particular place near Cape Town cultivate many useful products of the vegetable kingdom, including melons, that is to say the sweet melon, the honey melon, and watermelons. And Thunberg knew the watermelon from his teacher's description of watermelons that Linnaeus had received from Italy. Of course, in Sweden, they don't have either honey melons or watermelons. And so when he collected, he found various South African plants, and in the next watermelons or relatives of watermelons. And in the next 4 slides, I will quickly show you them just to get an impression. It's a very small genus, there are only 7 species. So here is a South African relative of the watermelon. Here's another one. This one doesn't have tendrils because it is lying on the desert floor and really doesn't have, there's no selective advantage of climbing on other plants because there are very few other plants. Here's another one. And here's another one. This one is also introduced in India. And here's the one that Thunberg was most interested in. He named it Citrullus lanatus because the young fruits have this woolly pubescence. And he thought it was different from the cultivated watermelon that Linnaeus had described, as I said, from Italian material, because it just looked different to him. Of course, he had very limited knowledge of these plants, okay? But he did think it was different, why he called it lanatus. Anyway, so when this student came to my lab to work for his PhD, he actually worked on ant plants in Fiji. This was a side project. I had been interested in this watermelon business there, and I said, okay, why don't we try and sequence some material, a little fragment of this plant collected by Thunberg, and a real watermelon edible watermelon, such as the one that Linnaeus described, and see what we see. And we found that even with the standard basic DNA markers, ITS and tRNL, really basic stuff, they were drastically different. This thing is not a close relative of the watermelon. So Thunberg was absolutely right and considering this a separate species. And people had fused these two because from morphology, as you can see here, you really have very little to go on. Nobody preserves the fruits. There's usually no data on the labels about the details, and the flowers also usually crumbled up. Morphology is terrible in its genus. So what is the archaeological evidence for the earliest human use? And why am I asking this question? Why not continue just with sequencing more and more examples of potential close relatives, which we've also done? Why do we also need archaeological evidence? Well, think for a moment, if you were to understand, let's say, the history of the iPhone or some smartphone, okay? Would it be a good idea to sequence humans and get the genealogy of, the humans that use the iPhone? No, you wouldn't get the history of the iPhone from sequencing the people who are using the iPhone, because people use plants quite independently from where these plants occur. The genealogy doesn't give you the use. You really need other types of evidence in addition to sequence data. Yeah? Because people share uses without being genetically related. Okay? So what's the evidence for the artifacts? What do we have? So we have some old seeds. And most interesting to me initially was these seeds, because they come from the tomb, the very famous tomb, the tomb of Tutankhamun, which was really one of the discovery of this tomb really opened up Egyptology, basically. This was discovered in October 1925 by this guy, Carter is his name, Howard Carter. Of course, these photos, this is an original photo, but this was colored after 1925. These are all black and white photos that people colored later, and we know that the pants have kind of a funny color. Anyway, I want to show you a very brief, 80-second video from foods found in the chambers of Tutankhamun, okay?
And lived there forever. But what did Tutankhamun expect to find when or if he got there? For Egyptologist Salima Ikram, the answer lies in some of the smallest items discovered in his tomb.
You needed to eat in the afterlife, and Tutankhamun was a teenage boy, so clearly he wanted to be well supplied. And so he had bread, he had wine, he had pomegranates, little green onions, and garlic, so he could flavor his food or munch on the green onion. And there are dates. There are piles of dates here. But what's really amazing about Tutankhamun's dates is that they were pitted, so he wouldn't have to chew around and throw out the pit. So these are really posh dates for the afterlife.
And these were just the appetizers. For the entree, Tutankhamun could choose from an all-you-can-eat buffet of Egypt's finest meats.
He had ribs, he had large joints of meat coming from the foreleg of cattle. And there are all these boxes of mummified poultry. Often the containers are made in the shape of what they are supposed to contain.
It was a feast fit for a pharaoh, pre-cooked, pre-packed, and
okay. So
lived there forever.
I wanted to end exactly on the basket where I cut this movie, on the basket that you saw, because in these baskets, 11 baskets like the one you just saw, is where they found seeds of watermelon. Okay, so it's very clear that watermelon seeds were in Egyptian tombs as gifts for the afterlife, for people to use in the afterlife, as we just saw. And from sandstone reliefs, such as this one, we also know that there are fruits that might be watermelon, such as this one, okay? And when I got a color photo of this exact tomb, the original publication is in black and white, it seemed like even more convincing that this would be a watermelon, especially since the items next to it. These are lotus flowers, and here are some snake melons, and here are some grapes. So the items were clearly like the dessert type of food, you know, sweet stuff to eat. It's to snack on or to eat after a big meal of meat. So the sources for inferring the history of the watermelon are summarized here. So this watermelon that we just saw, it comes from near Cairo. Another wall drawing from another tomb near Asyut. And then this preserved leaf that is preserved in the museum in Kew, Royal Botanic Gardens, Kew. They have a special museum for economic plants. And then some seeds not so well preserved, discovered in this Nubian city of Amara. This is in northernmost Sudan. Okay, and then some ancient seed that you actually saw on the earlier slide from Libya. All right. So it seemed, let me go back. It seemed that this would be the easiest to get DNA from. Okay, leaves usually give, you know, good DNA. Why would you bother with 6,000-year-old leaves when you could start with an ancient seeds, when you could start with an ancient leaf? And so this is, of course, what we started with. I emailed the curator of this economic plant museum there at Kew and asked him to open the box. You see here this when he just opened this box, which hadn't been opened since 1888, and got out a fragment and sent it to Munich for us to sequence. This leaf was actually collected by a German guy, George Schweinfurt, in this necropolis. Okay, it's many tombs. That's exactly where he worked. He worked there in the 1880s, and he, as a botanist, and he was fluent in Arabic. You know, he was really experienced collector and scientist, and he opened many tombs with others. And he writes in his notes that he got watermelon leaves that had been used to stuff one of these tombs, not this exact one, not tomb, a sarcophagus. Okay, and this is all well documented. So I had no reason to have any doubts that this was actually a leaf collected from a sarcophagus by this guy and sent to Kew, right? So with much effort, one of my colleagues went to Berlin to a special DNA lab. She was properly dressed to, you know, make sure that the valuable piece of leaf wouldn't be contaminated with modern DNA. And then Oscar, another student who's working at the Royal Botanic Garden, did the bioinformatics work. It was all wonderful. We get excellent DNA, wrote the paper and submitted it to Current Biology, and it was in review when, because of another project, I sent some of the leaves, some of this leaf to a special lab in Switzerland to have it C14 dated. Okay? And so here's a fragment again. Here's the fragment we had. We sent it to Zurich, and it was dated. This is a C14 dating to 148 years before present. Okay, so 148 years before present, before the 1880s when Schweinfurt collected it, that would have been before present is in 1950s, excuse me. So the 1950s is the date which is considered present in C14 dating, okay? So that minus 148 is 1870. 1870 is around the time that Schweinfurt worked in Cairo and collected the leaves from the mummies. And he probably got some modern, at his time, modern contemporary leaves for comparison, okay? So he didn't lie, he did find leaves in the sarcophagus, and he did compare them understandably with, you know, modern watermelon leaves collected nearby. It must have been something like that. And then they were sent to the Royal Botanic Gardens Kew and put in this wonderful box and considered ancient leaves by some mistake, completely understandable. But it was terrible for me because we had to retract that paper. Luckily, it was in the second round of reviewing. It was really incredible, and it really taught me a good lesson. But if you want to work with ancient DNA, you have to do the C14 dating first. Anyway, so we had these seeds. We had the Tutankhamun seeds, okay, a few of them. We had some Libyan seeds, and I had this great American bachelor student, it was her bachelor thesis, who was interested in 3D reconstructions. In fact, she did her master's then in 3D reconstructions of a completely different zoological organ, a snail actually. Anyway, so she was interested in studying the morphology of these seeds, which I thought was a good idea to see whether one can even identify these ancient seeds. And so we compared them using the best possible, computer-based tomography with all the modern watermelon seeds, which you see here. And we became more and more doubtful that one can even be sure about the species you are dealing with. Yes, they're watermelon seeds, but whether it is species X or species Y, who knows? And also what struck us were these damages that we saw, okay? And the damages are probably explained by people eating these seeds, because when people snack on watermelon seeds, which they do even today, they cause characteristic cracks. And so this could be an indication that people 6,000 years ago were snacking on these seeds. But as I said, I was very careful now about really dating the seeds before even worrying about any ancient DNA. Before I show you the dating and the results of the dating, I want to show you another very brief 60-second clip that shows the landscape where these seeds were found. It's a very famous archaeological site.
The memories of Saharan grasslands, rock art sites of Tadrat Akakas.
That's where the seeds were found.
Tadrat Akakas is a mountain range located deep in the Sahara in southwestern Libya. Over 10,000 years ago, this area was covered with rich vegetation and inhabited by wild animals and humans. The images carved on these rocks portray some important evidence. This is a rock carving of a rhino. Rhino usually live in grassland and love to bathe in water. This is a crocodile, which also lives near water. There must have been some rivers and lakes here.
Okay, so clearly the vegetation at that time, 10,000 years ago, and until 7,000 years ago, was much more humid. This is known from many studies. And this site where our seeds were found is actually super famous. It is called Uanmajuku, it doesn't matter, in this rock shelter that I just showed you. And what they found there was baskets, they are C14 dated, and the seeds that I showed you, and most famously the so-called black mummy. If you Google black mummy, you immediately come to videos and documents about it. This black mummy is dated to 5,500 years before present. It is beautifully preserved. It appears to have negroid features, and the most important thing, it is about 1,000 years prior to when the oldest Egyptian mummies preserved in this way are dated to, okay? So these people living there had mummification techniques that apparently were of the highest standard much earlier than the Egyptians. Okay, so it's a really important site. And so I actually did receive 4 of these seeds from the woman who had originally discovered them in the 1990s, and she gave me permission to isolate DNA on the 5th of December 2018. But now I had become smart, as I mentioned, okay? We used 1 seed for C14 dating, 2 seeds for the computer tomography study that I just showed you, the study of morphology, and 1 seed for the DNA isolation. I never got permission to isolate DNA from the Tutankhamun seeds. It's too bad, because of course we would have loved to do those too. So the question then was, were Stone Age Libyans eating watermelons 1,700 years before the Egyptians? Because based on morphology, this was called Citrullus lanatus, the watermelon. And what we found, and this is the paper that just came online, was that here, let me just orient you, okay? Here's that Schweinfurt leaf, and it's just the genome of that Schweinfurt leaf is just a modern watermelon. These are modern watermelons, okay? And here is the ancient genome. the Libyan seed that I showed you, and also another one which we did, I didn't talk about this much because it's so poorly preserved, from Sudan. It's 3,000 years old. This is really C14 dated, okay? And what we found was that these ancient genomes were not modern watermelons, not watermelons as we know them now with red pulp and sweet flesh, but they were instead closest to something called mucosospermous. I will show you pictures of this in the next slide. Okay, and this mucosospermous, it has a western clade in blue and an eastern clade. So this would be West Africa, and this is East Africa. And here's our ancient seed. It is clearly belonging to this mucosospermum entity. And the other younger DNA, this one here, actually gave less good DNA, and its genome was not as informative as the older one, which was a surprise because it's twice as old. Okay, but what you can also see here, that there is admixture. Okay, the pure West African thing is in green here, and there's some admixture, and both our ancient genomes also show this admixture. I'm not going to go into this in this talk. Okay, so what is mucosospermum? Here is mucosospermum, this West African species. It is only used for its oily, nutritious seeds. It's very important. If one Googles this, there are many, many recipes. The pulp is not used. It is very bitter. So it appears from the DNA that these Stone Age people in Libya and that rock shelter were actually using the seeds, which fits with the damage that we found. Okay, so it could be that they were snacking on the seeds, not on the pulp. And genetically, that plant is closest to the West African mucosospermous. This leaves the question that still has to be answered: where does the sweet pulped watermelon come from? The one that was then domesticated and is now eaten all over the world, especially in China. So the Russians in the 1960s had collected material at the border between Egypt and Sudan. Okay, Sudan at the time was communist, as some of you will know. It was oriented towards Russia. There were like 20,000 Russian advisors there, especially military advisors, and also Russian botanists. And they collected these watermelons at the border between Egypt and Sudan. And they brought them in cultivation in southern Russia. Okay, they really measurements on the sugar content. These have sweet pulp. It's not super sweet, but it is sweet. And so it is plausible that this could have been the precursor. And last year, we published a paper, another paper, it actually came earlier, on a high-quality genome obtained from material that I got from Sudan. Here, I got this from a friend in Khartoum, of this presumed precursor. Okay, 2 genomes were from the USDA agricultural collection, where we got seeds and then produced these. And then this is our high-quality genome. Okay, and these are the old Russian photos from the 1960s. And here you see all the 200 some modern watermelons. Okay, many of them, of course, Chinese. And here you see that presumed sweet-fleshed precursor. Here, those 3. Okay, and here again you have that West African mucosospermum stuff. So really, this one is closer to the domesticated watermelon, and very likely was the precursor from which modern watermelons were domesticated. So I'm almost at the end. What does DNA, ancient DNA, contribute? One is the identification. These old seeds, based on morphology, you really couldn't know whether it was this or that Citrullus. So that is the one thing. But the second thing that DNA contributes is the traits. You can infer the traits of interest. This is so wonderful. So many of these watermelons, let me go back. Many of these things that you see here, all the ones here in purple, okay, are actually from herbarium specimens. Herbarium specimens collected between 1824 and until today. Okay, these are all herbarium specimens. And we can know from the genome whether they have bitter flesh and whether they have, or sweet flesh, and whether they have white whitish green pulp or red pulp, because the particular mutations are very well understood and we can see them in the genomes. Even in that ancient genome, we had enough coverage to really detect that particular single mutation in the amino acid so-and-so that causes the loss of bitterness or that causes the red color. And so we were able for every single sequenced specimen to infer whether it was bitter or sweet. And this was also possible for this ancient plant. You can see it had greenish white flesh and it was bitter. Here's an interesting mutant, okay? Sometimes, of course, there will be a mutation and there will be a loss of bitterness in a wild species. So this brings me to the end of my talk. So here's the story of the watermelon. The West African seed use watermelon was apparently the one, or very close to the one that the Stone Age Libyans were snacking on. They were clearly not eating the pulp because it was bitter. The one that Thunberg collected in South Africa is not close to the sweet flesh watermelon, nor are any of these other South African ones. The Egyptian tomb paintings and also the tomb content, for example, the seeds in King Tutankhamun's tomb, suggest that Egyptians ate sweet desert watermelons, such as the one that were painted on those trays, and also snacked on the seeds. So they used watermelons in both ways. And the Kordofan watermelon, this is really a subspecies, was very likely the direct precursor from which Nubian or Egyptian people, probably the Nubians, because this is Nubia here, were domesticating the watermelon. And I want to end with a comment. Our team consisted of 2 Colombians. The 2 first authors on this most recent paper are from Colombia, a Pole, 2 Chinese, 2 Brits, 1 French, and 3 Germans. Of course, most of us never met, only on Zoom. And very sadly, we don't have an African collaborator. I tried, but so far I haven't found an African collaborator to continue this work. Thank you very much.
So, thanks, Suzanne. It's amazing how you can combine different disciplines, right? It's just extremely transdisciplinary work.
Yes, yes. And people were surprised. I tell you, it wouldn't be possible without people sharing all this stuff and information. Just super.
Yes, exactly. And here at the university, we discuss a lot of how to do transdisciplinarity. And that's not so trivial. To connect the people to, you said that you couldn't get authorization for sequencing some samples.
Yes.
And thinking about the reality in South America, that it's even harder for us to get in contact, to receive material via mail. So
yes,
how would you suggest us to think about, and here, thinking about domestication here of the Andes, and that is a center of domestication. But I was discussing with a person the other day about domestication of cassava. There are some new papers on it, but we don't have this ancient material. We have just some small phytoliths.
Yes.
Do you think it would be possible to sequence phytoliths or things like that?
No, phytoliths, I don't think so, because they are also chemically, they don't have the DNA.
They don't have DNA, right?
But you have, surely there is so much in the museums, and the question, as you just said, is the sharing. You started when you introduced me talk about women in science, and actually it was so interesting. The people who shared were all women. The Egyptian government did not give permission to sequence the seeds from Tutankhamun, even though they have 11 baskets with hundreds of seeds, hundreds, okay? They didn't give permission. I'm not saying that these are necessarily men, but the people who did share were all women. The woman from Poland who sent me the 4 seeds that she had dug up there in Libya in the 1990s, she just sent these to Munich and gave permission just like that. And the other one, the Amara, was a woman, okay? And many of the color photo was a woman who sent me this, because when I saw the color photo, it really looked like a watermelon. When you see these black and white old photos, you never interpret it as a watermelon.
Yeah, really.
It was so interesting to me that it was women who are less, maybe less.
It's easier for us to give things, right?
I think so. It's very funny.
And you started your talking explaining a bit that, okay, it was a PhD of Chomsky, right? And this was a side project of idea that you had.
Yes.
Can you just please let us know how did you have this idea? I know that you are interested in domesticated plants, but how to combine how to think, because there are a lot of taxonomic work.
And you know this also from your own PhD work on the papaya. One has to be so skeptic of old morphology-based identifications. I have full respect for the taxonomists working with this dried material of, they could not possibly distinguish these species based on that dried old stuff. I have full respect, but one has to be very skeptic and get a little fragment and sequences. Yes. Now this work is very expensive. The high quality genomes are super expensive, and so you can only do it, I think, if it is an economically important plant. Remember when you and I were discussing possible PhD topics, and then we ended up with the papaya, which was a good choice. It has to be something economically important. And so my little side project on Citrullus, I was really thinking about Citrullus is great. It is economically important. It's a small genus. And initially I wanted to go to Africa. Guillaume and I, we actually had DFG money to go to Sudan, but it was too dangerous. And so,
You gave up the idea.
Oh yeah, so we only used the old dry herbarium material and the seeds that my friend from Khartoum sent us.
Yeah, but I understand you about this taxonomy, because here in Brazil we have I feel that we have more investment in taxonomy, basic taxonomy, than in Europe, right?
Yes.
And I think when you say about, okay, we can't go there to collect, we need to the specimens, and even that we can't really believe on the identification only on morphological things. How do you think the taxonomic work, helped you and your knowledge about taxonomy, because you have a huge experience on it, mapping things, reading things.
Thank you for the question. Yeah. It's an excellent question. Without my taxonomic knowledge and all this old literature on this collector so-and-so going here and there, people wouldn't have known, my students and my collaborators, all these 7 collaborators, they wouldn't have known which herbarium to go to, which place, exactly. That old stuff that Schweinfurt collected, blah, blah, blah. You need the old herbarium-based understanding, for sure, to even know where to go.
Yes, even about the plants, the distribution, the geographical distribution. How do we know that without taxonomy, right? Basic taxonomy.
Yes.
Because it's sometimes I just feel a bit, okay, taxonomy, and shall I go back to my monograph of papaya, right? So of Paricacea, yes, I should go, because and when I look, I heard you. Okay, I go back to my Paricacea.
Yes. But on the other hand, I have learned so much from this interaction with the more molecular scientists, because what you get from these genomes, like I said, you can really infer the trait of the plant. Of course, also the other traits. I just focused on bitterness and flesh color because it's so easy. You can also infer traits that you would never know from morphology. Okay, and also this whole thing about hybridization, which I didn't go into because it's just so difficult. This hybridization, you would never see it in the morphology.
Yeah. So we have some questions or not. Let me see here. Okay. Just a minute. Because I still have one question for you, but
Take your time.
okay, the comment here.
Late.
Okay, we have a question here. What was the main challenge sequencing a very old sample?
Oh my God. It was we were lucky, okay, that the 6,000-year-old seeds gave a better genome than the 3,000-year-old one, okay? So in a way, we were incredibly lucky. So the main challenge is really having professional collaborators. This has to be done in a lab that is dedicated to ancient DNA. That is not something you do in your own lab. And I was very lucky that we had these collaborators in Berlin, south of Berlin in Potsdam. This lab of, his name is Anton Hofreiter. He is constantly in Nature and Science. Just, I don't know, 10 days ago with the history of the dog, the history of the wolf, the history of the mammoths, stuff like that with others, okay? Not just him. It was all animal DNA because it's much easier to get than plant DNA. But this lab is dedicated to doing nothing but getting ancient DNA. And it costs a lot of effort to get enough out of that old seed. You have one old 6,000-year-old seed and some fragments that are 3,000 years old. So the main challenge is the getting the good DNA, and of course the bioinformatics work also.
Yeah, because it's not just old, it's just very few amount of material to extract, right?
Yes. This is not something you do easily, but not everybody has to use 6,000-year-old stuff. You could use 500 years or 800 years, which is already, of great interest. Even the herbarium material from 1824, because people started really working with genetics in the 1950s, with breeding, targeted breeding. The modern watermelon breeding started in 1910 and really in the 1950s. And this really the high modern papaya, I'm sure, is also from the 1950s onward. So even if you have plants from 1700 or a fragment in some museum, it's of great interest. It doesn't have to be thousands and thousands of years.
Yeah, that's interesting point. Opens the mind for other questions on
what you have in Belém,
improvement, right?
Yeah, in Sao Paulo and in Belém, and I'm sure you have a lot of Indian fletchers and baskets and lots of plant-based stuff that could be used, and it's maybe 250 years old.
Sure. That's true. Yeah, that's true. Well, we have a comment here from Miguel Echeverria. This was a really interesting, great presentation. Thank you.
Thank you.
So just to finish our talk here, I would like to, as a leader in science, we know that you are one of the greatest botanists in the last 50 years here in the world. So, and when I hear you are talking about money, how expensive it is, how the structure we need to do the kind of research, and how the academic world is driving us against the cheap research, right? Yes. That is the field work and the basic science, and that we can't publish it in high impact factor anymore. And we are moving in this way. It's our money, right? The impact factor, number of papers. And we have to deal here in Brazil. I'm talking about my reality here. We have to deal here with a lot of bureaucracy. Well, I think you also has this bureaucracy, but the money that's going out from science here is running out. We don't have so much money. How do we? I understand that we have to publish our results. It's important, but how would you think now that you are retired now and you are more relaxed? I think. What do you think about the way we are organizing the academic world?
Okay,
this point of view that how can we balance realities of those who has a lot of structure and a lot of money with those that doesn't have it?
Yes, yes, yes. So my one answer, and I think you suspect this answer, coming answer is target important plants. There are, 250,000 species of angiosperm, something like that. We have a choice. We have a choice. And there are species that are of greater importance for mankind and species that are less important. Okay, and in Brazil, of course, you have so many important plants. Important for humans because we have to target the brain power and the money and the facilities, the labs. We have to target them to plants of interest. I really think that's fair enough. It is fair.
Yeah,
I think it is
true.
okay. I do think it's possible.
It is the possibility, right?
Right, and society, I mean, this money comes from the taxpayers ultimately. Okay?
Mm-hmm.
The money for research comes mostly from the taxpayers, at least, yeah. And I do think it's fair to say that we should focus on important plants. Of course, then they have unimportant relatives. I know that.
Yeah.
I think applying one's brain power to important plants, I think, is a key thing. And I didn't follow that advice. Early in my career, I worked on lots of unimportant plants. It doesn't matter. Lots of unimportant plants. And I wished I had focused on ones that are of interest to people. I see this much more now. We don't have the luxury to work on, we will never describe all plants. There's no chance.
Yeah. Well, okay. Thanks a lot.
Thanks for nice seeing you again.
Nice to see you, and let's keep in contact. How do we finish here, girls, please? Yeah.